Okay, so this will be chapter 14 in the textbook. If you're interested in reading the textbook, remember you can go to Cell Lounge, right? The new codon assignments that the first one due tomorrow night at midnight is on genetics. And we're going to talk about Mendel and classical genetics. All right, we're going to start talking about a little bit of information that will give you the foundation so that you can do well in the genetics class that you'll take typically as a sophomore. Everybody has to take in biology. Mendel studied peas, and that's what this picture shows you is a pea plant, and this is the flower that he studied, and he is known as the grandfather of genetics. So, the only thing I'm really going to talk about today is just to make sure that after all of this stuff that we've done so far, particularly cells, organelles, nuclei, we're going to stick to eukaryotes, that remember, life exists because of the manual that we have, right? And what that manual is the genetic code, and it's stored in chromosomes, right? And we have more than one piece of chromosome. We have 23 pairs of those. And on those chromosomes are hundreds of gene locations, thousands of gene locations in some chromosomes. And those locations, which we call genes, have a name. They're called a gene locus, all right, which is on the next slide, but I'll just write it down now. So a locus is the location of a gene. And you could go and look up where the gene for hair color is and on what chromosome. Aspect of an arm on a chromosome, it'll give you a number, it'll tell you exactly where it is, right? The genes that we have mapped. And so this locus is a location for a gene. And what a gene is, remember, is a functional region of DNA, a sequence of DNA, that codes for RNA, right? And the RNA that we're interested in here is messenger RNA because that carries the message for protein synthesis. Now, we know as a diploid organism that we actually have two copies of each gene, right? One from mom and one from dad. Right? And those copies can be slightly different from one another, variation, which we call alleles, right? So alleles are variations in the instructions at a gene. Red hair, blonde hair, right? I'm a redhead, my wife's a blonde, all my kids are blondes. No redheads. Because her copy is dominant to my copy, right? Or is the one that is expressed. So we know every cell receives a complete copy of each chromosome, and if you're a diploid animal, you have two copies, one from mom and one from dad, right? All cells have the same genes on the same area and the same location of each chromosome, and that includes the copy from mom and the copy from dad. And the central dogma is what runs everything, right? As far as cell biology goes. So the DNA that we're talking about here is nuclear DNA, which there are several feet of, right? And the process of copying a gene locus for its genetic information or its instructions is called transcription, right? That produces messenger RNA, which is single-stranded. Messenger RNA can then leave the nucleus and find a ribosome, where it is then translated into a protein, which we call a polypeptide, right? So slight variations in a gene can change what the polypeptide looks like, right? That is, we can maybe replace one amino acid with another amino acid because our instructions are slightly different. If we change too many of the amino acids in a polypeptide, it can change the shape of the protein. And we already know structure equals function. If you change the shape of something, then it's going to change its function maybe a little bit, right? And that's kind of how genetics works. So we're tying molecular biology, cell biology, and genetics together here. Dogma to what we're talking about. So remember, we're going to translate kind of what a gene sequence is, right? Because it codes for a protein, and then some of those proteins are going to give rise to features we can see externally, for example, or internally, that we can call a character, like eye color, hair color, blood type. Those are characters, right? And then those characters, as we're going to see, have variation in them. Usually at first just one or another variant that we're going to call a trait or a phenotype. So those are linked to the protein and to the central dogma. So what sequence you have in your DNA leads to a copy of messenger RNA that leads to a protein that affects what we see in an organism. So, use these terms before. Again, lots of terminology you've got to keep straight because I just start throwing the terms around, and I always assume you know what they are. Yeah, we've gone over. Yeah, this is just, again, central dogma, showing the same thing that last slide just showed, that our genes are going to be in our chromosomes, our DNA, messenger RNA, and protein synthesis, so central dogma. This one just shows it a little closer up, showing you the actual DNA has a sequence of bases that code for that protein of interest, okay? Well, now I'm going to show you another slide here in a minute that shows you how this sequence can look different and lead to proteins that are different. And that leads to our traits that we see. I'm glad you said that because I came back. I don't know if I mentioned this. Gene expression is the process of converting our instructions into a protein. So what do I mean by that? That means if I'm producing a protein and I'm a cell, then I'm expressing that gene. So just because I have a gene doesn't mean I'm going to always express it. Does that make sense? Sometimes you might control when you express it. Like you turn it on under certain environmental conditions to produce the protein of interest. So when we're expressing genes, or when a cell is making proteins for a gene of interest, we are doing gene expression. Make sense? Okay, a locus versus an allele. So every gene that we're interested in that codes for a protein has a location, and that location is consistent across an organism. And so it's just a location of a particular gene on a chromosome. So remember, what does that mean? That means it's somewhere on a piece of DNA, right? And we have a certain number of pieces of DNA. We have 46 of them, right? 23 pairs that can condense down into chromosomes. On those chromosomes, they locate sequences of DNA that code for something. And so they might say the long arm of chromosome 21 at this location is the location for the character we call eye color, for example. And then at those locus, or at those locations, I should say loci, we can have alternate versions of the code or the instructions, which we call alleles. So when we get to understanding how Mendel did his experiments, the first thing we're going to realize is that the characters he picked have two alleles, right? Or they have the outcome of two different versions that lead to two different traits: purple color, white color, right? So those allele alternatives, or those different versions of the instructions, lead to a different, what we're going to call phenotype, or what we see externally. Now, sometimes alleles are called silent alleles. Or sometimes they're called neutral alleles. These are alleles that you have a little alteration in the code, but it doesn't do anything to the protein. The protein still functions properly. Does that make sense? And so, for example, you might have 20 bases that make up a gene sequence, and you change one C to an A, and it does nothing to the protein. The protein still functions properly. So those are called silent or neutral alleles. Sometimes you might have minor or even major changes to the protein function. Because remember, the sequence determines the amino acid sequence, right? The sequence of bases in the DNA is going to code for the sequence of amino acids and how they look in the protein, which controls its three-dimensional shape, right? So if you alter the instructions too much, the protein is going to change shape. And if it's an enzyme, it's not going to work right. Does that make sense? So some of these allele versions are silent, some of them are minor changes, some of them are really large changes that you see. and they may alter whether the protein functions properly or not. We'll see this with flower color, and I'll show you a picture of why there's a difference between purple and white. It has to do with whether the protein's functional or non-functional. Remember, and we've mentioned this many times, diploid organisms, like us and plants that we're going to talk about, inherit at least, and for us to keep it simplified, 2 versions, right? Or 2 alleles. Sometimes they can be the same, and sometimes they can be different. And we get one of those from each parent, right? And the diploid organism is what dominates in our diplonic life cycle. That realization by Mendel, he called the law of segregation, okay, which is on the next slide. So Mendel's first law that he came up with, it's called a law because it's pretty much set in stone. It's pretty much as proven as you can prove in science. That organisms that are diploid inherit 2, he called them units. Because Mendel lived in the 1800s. We had no idea what DNA was in the 1800s. So using mathematics, because he was a mathematician also, very good statistician, he worked backwards from his data and figured out that in order to get the results he's seen, these plants have to inherit 2 versions of whatever is controlling that trait that he's seen. So he called these inheritable versions units. We now call them alleles on chromosomes, right? And you get 1 from each parent. So when 2 parents breed, those 2 units come together in the offspring and will give you a certain trait. And then that offspring is going to put those 2 alleles in separate gametes during mating. So there's a maternal unit or a maternal allele, and there's a paternal or dad allele, right, or unit. And then those alleles segregate independent of one another, law of independent assortment, like we've talked about, in different gametes during meiosis. So here's one of these examples that Mendel used. I'm going to give you the 7 major ones that he used early on in his experiments. But here's one up here. These are peas, and there are 2 traits to the character of pea shape: smooth and wrinkled. You always get either smooth or wrinkled. And so he always picked these very discrete traits that had 2 versions, if you will, right? Which were controlled by 2 alleles. And so here's what it would look like on a chromosome. Flower color is another one. So here's one from mom that carries the purple version of instructions. Here's one from dad that carries the white color for flowers. Just a very small difference in the instructions. So far, so good? All right, so genotype. We again defined these in the lab last week. So genotype is the combination of alleles you inherit from mom and dad. We're going to give them letters, all right? And that's how you're going to do these you already started this, right? You've already done this in the lab, and you'll do it on the test. You give letters to your different alleles if there are different versions. And typically 1 is going to dominate, and we call it the dominant or wild type. You often see wild type also used for this because it's the one that typically shows up most often in the wild if it's an organism that lives out in nature. All right? And the dominant allele is going to mask, whenever it's present, the recessive allele, which means your heterozygotes, one with a big A, or the dominant allele, one with the small a allele is going to mask so that you don't see the recessive trait. Now we also have these terms homo versus heterozygous. Homo means same, zygous means allele, okay? It means zygote actually, but in this instance it's allele. So they have the same version for each of the paternal and maternal alleles, and they're dominant. That is, they would mask the recessive. So we're homozygous dominant. Or you can be homozygous recessive, where you would carry both recessive alleles. Or you could be heterozygous, which means different. So you got a big A and a little a, right? Alleles. Now these genotypes will appear as a trait, or what you see. And those traits are called phenotypes. So trait equals phenotype. Character is what you're coding for. Flower color, height, eye color, hair color. Those are characters, and you have different traits or different versions of those characters, which we call phenotypes. So notice your phenotype here, your phenotypic ratio, or your phenotypes are either purple or white. And you're purple if you're homo or heterozygous dominant, and you're only white when you're homozygous recessive, right? When you're carrying both of these alleles, and you can't be masked by anybody. There are other flowers that do this that are red and white, or another color and white. We'll also talk about one of the differences when you get red, pink, and whites, right? Which we call incomplete dominance or mixing. Okay, everybody good with genotype and phenotype? It's Monday, I know. Are you all alive? And it's raining. We need the rain, right? Now, I'm visual, right? You know this already. Kind of said it 1,000 times. So I like to show you pictures of what it looks like meiotically. Now, since we're only following one gene here, we only care about one chromosome, right? I mean, even if this pea plant has 100 chromosomes, we really don't care. We're following one chromosome, because that's the only locus, right, for this gene of interest. So you can see here, here's homozygous dominant. Here's a homozygous recessive germ cell, right? Remember, germ cells become gametes. The first thing we're going to do with our germ cells, or our developing gamete, is to undergo synthesis, right? Going from single-stranded to double-stranded chromosomes. This is one homologous pair, the only one we care about, because it's the only one that's carrying a copy of mom and a copy of dad's gene of interest, the locus for that gene of interest. So notice when we duplicate the DNA, the homozygous dominant now has 2 copies of the big A chromosome, right? We'll go undergo meiosis 1 to form 2 primary spermatocytes, right? It's what we're forming here. Or actually, this would be a secondary spermatocyte now, right? That would have 1 chromosome, the 1 chromosome of interest, which is double-stranded with our big A's. And then we go undergo meiosis 2, and notice we're segregating, law of segregation, each individual allele into a gamete, right? For this one, it's pretty easy. They're all big A's. So our 1 germ cell became 4 gametes, whether it's egg or sperm, with the big A, right? A single-stranded chromosome with the big A. Same with the homozygous recessive parent. They're all recessive. So when we breed these 2 parents, there's only one possibility, isn't there? For this particular gene, and that is a heterozygote that will have 1 of mom's and 1 of dad's chromosomes, correct? So it would be homozygous dominant. Good? So there's a little sperm swimming to the egg. Oh, heterozygote, right? So you get your alleles from your parents, who got them from their grandparents, who got them from their great-grandparents, and so forth. And so those, you can trace those heritable traits in a family tree, which you kind of did with your Martians, right? We went backwards to figure out, who everybody was. You can do that. That's what genetic counselors do, is trace rare disease states that are controlled by genetics, for example. So this is one of my favorite pictures in the whole lecture, because it puts everything together, kind of applies what we've learned to this point. So we've already talked about these 2 chromosomes and this locus for flower color. And the way flower color works is pretty simple, actually. The 2 traits you see are based simply molecular biology-wise, or what's happening in the cells that control color in the flower, is that the purple allele has the right instructions. The instructions actually produce a protein that works. The enzyme actually can produce purple color. That's why you see purple. The white flower, you change 1 or 2 bases. In this instance, it's showing 1 base change, a C for an A, right? Simple mutation. happened sometime in the evolutionary history of this plant. This protein doesn't work. You change one amino acid and it doesn't work. It doesn't fold right. So the enzyme doesn't work anymore. If it doesn't work, it can't produce purple, it's white. So that's why you see purple and white, or red and white, or beige and white, whatever the color of the flower is, okay? This is pretty rare, just not, it's a pretty rare mutation, or pretty rare occurrence within a population. So the dominant or wild type purple color is what you often see. You see white more rarely. So can you see here that genes can mask or hide other alleles? Just because you have an allele or an instruction difference doesn't necessarily mean you're ever going to present that, right, during your life. This is how mutations that are bad hide. Because you're fine, but you can pass a gene on that's recessive to your offspring, that if your partner has the same recessive gene, a baby can express something you don't have. Does that make sense? You can hide disease states, you can hide other alleles. So this is simple dominance and recessive genetics. Okay, so Mendel was smart. The first thing that Mendel did when he started messing around with plants was that he picked very discrete traits. He picked characters that had this or that. There were just two possibilities. There weren't 3, 4, 10, 12 variations. There were only one or the other. And so he picked 7 of these. And in order for us to understand kind of what he did, we have 3 different generations that we're going to keep track of. We have the parental generation, which he picked on purpose because they either have this or that, right? And he bred them. All right, and we're going to call that the parent generation. The first generation of seeds that he then grows up, we're going to call the F1 generation, or the F stands for filial, which means son or daughter or offspring. The F1s, once he gets them grown up, he's then going to breed them to each other to get the F2, which is the second generation of kids from the F1. And then he will grow those up and look at them. And he's looking at specific traits, right? So for this first one, it's flower color. So he has purple and white flowers. And the thing is, this garden that he took over at this monastery that he was the abbot for, and then he did all these experiments at, they had been breeding peas for a while. So they knew that, hey, this purple plant always gives rise to purple plants. This white plant always gives rise to white plants. And the reason that pea plants are so important to understanding this is because plants can self-pollinate. All right, do you guys understand what that means? They carry both gametes. So if I'm a white flower, I'm carrying white sperm and eggs. And so if I just let that flower make new babies, they're always going to be white, right? Same with the purple. It's always going to be purple. It's only when I breed them, make them crossbreed, a purple and a white, that you'll start to see maybe some differences. So you can control that. How do you do it? You remove the organ on the flower that makes the pollen, right? Because pollen typically flies away or is carried away by bees, for example, where the egg is encased in the middle of the plant in something called the ovale. So he was able to go in and take anthers off and then act like a bee, take some pollen from one and add it to another flower. And then he could produce his own types of artificial offspring, if you will. So what he did is he took what we call pure breeding, which is another term for homozygous, a pure breeding dominant flower, purple, and a pure breeding white flower, white, and he self-pollinated them. Well, he didn't self-pollinate, he cross-pollinated them, sorry. And we end up with the F1 generation of all purple flowers. And he didn't do this like 8 times or 9 times. He grew up like 6,000 to 8,000 offspring. All right. So this isn't just like a flip of a coin kind of chance event. He had huge sample sizes. He was loved plants, grew lots of plants, right? In this big garden. So he raised a huge population, or a huge group of, probably a better term, of F1s, and they were all purple. So he was like, the white disappeared. So remember, this is the first time he's doing this. And the question, of course, as a curious scientist is, where did the white go? Why aren't there any white flowers, right? And the reason for that is because you have a purple and white allele, and the white is now hidden or masked by the dominant trait. Now, he could have stopped there and been like, oh, wow, white is an aberration, a mutation, something that happens really rarely. But he took these guys and now bred them together, right? 2 F1s, and bred them together over and over and over again. And he saw that the white would reappear. Now, it wouldn't reappear very often, but it did reappear. So you started with it, and you get it back in the F2. The only possible explanation of this, if you work backwards statistically and trust your results, is you have to inherit 2 units, right? 2 versions of this in order to get one or the other version. And white reappears because obviously one masks the other one, right? It's actually pretty incredible experiment. It's very classical, very simple, but to work backwards and do that for the first time is pretty incredible, actually. He's why he's called the godfather of genetics. Now, when you look at these, you're going to get a different genotypic versus phenotypic ratio, which again, you practiced in lab. So notice with our little Punnett square down here, with our 2 gametes that we get from our 2 parents, we get 3 purple to 1 white, right? Or 75% of the time you see purple, hence why it's called wild type, because it's very commonly seen. And the white shows up about 25% of the time, or 1/4, right, of the time. So 3/4, 1/4, as far as percentages that you see. So it's 3 to 1, dominant to recessive phenotype. The genotype is 1-2 to 1, right? And he had to guess the 2 to 1 on the purples based on doing coming up with a simple Punnett square. You could figure this out by doing a test cross, which we'll cover later. And you kind of talked, I think you had a question about a test cross in those little genetics questions at the end, where you would have to breed these back to a homozygous recessive individual to show that they were heterozygous or homozygous for the dominant trait. We'll cover that in a minute. But you got obviously 1 homozygous dominant and 1 homozygous recessive, and then 2 heterozygous dominant individuals. That's where the 1-2 to 1 comes from. So 3 to 1 phenotypic ratio, 1-2 to 1 genotypic ratio. That's typical of any heterozygote for a single monohybrid cross for a single locus, if it's simple dominance and recessive. If you get something other than that, then something else is going on, right? So far, so good? Wild type is the dominant phenotype. Just another name for the dominant phenotype. I tell you that now because that's how in genetics they always say the wild type of Drosophila is red eyes, and which means that's the... Yeah, so it's just the dominant trait, right? Yeah. So these are monohybrid crosses. We always start off with true breeding. Again, another term. True breeding means homozygous. Isn't biology horrible? There's like 780 terms for the same thing. It's just ridiculous, right? So wild type, true breeding, homozygous, heterozygous. You got to keep track of all of them. So true breeding are homozygous. A true breeding individual can be dominant or recessive. That simply means they have 2 dominants or 2 recessive alleles, right? A heterozygote would not be true breeding. Does that make sense? Because you don't have the same allele present on your 2 copies. So true breeding individuals, 2 homozygous individuals that are dominant and recessive, you breed those together to get you're heterozygous, right? Your heterozygote would be your F1. So here is our example for you with basically what our seeds look like, right? Whether they're smooth or round seeds. The smooth is the wild type or the dominant phenotype. And so you get capital S. And then the wrinkled is the recessive allele. So if we take a true breeding smooth times a true breeding wrinkled and we breed those, right? You could take pollen from one and add it to the flower of the egg of the other. Remove the anthers of the one that you pollinate with your pollen, right? The pollen you collected, and you'll get all heterozygotes that are all smooth, right? They all appear smooth, but they're all heterozygous. That's the F1 right here, right? The F1 plants. He did this, grew lots of these for, again, the 7 characters. I think they said in his original 7 characters, he grew over 30,000 plants. That's a freaking ton of plants. I think he did it from, I think it was 1855-ish to 1863. So it took many years to grow all of these. Lots of growing seasons, if you will. The other thing that are nice about pea plants is you can store the seeds, collect them, start back over the next summer. So he did that. Now, if you take these guys, the F1s, and you make them breed together, then you're going to get 3-to-1 smooth to wrinkle. So for each of these characters, he did it, and he got the same results, this 3-to-1 or phenotypic ratio to 1-2-to-1 genotypic ratio. So here's a famous painting of him working in his garden. He loved his garden. He spent all the growing season in it. So he was born in the 1820s to very poor parents that were farmers. And so he loved plants. He raised plants, raised bees as a child, was very intelligent. They sent him to school. And, they had this kind of combination of what they call high school and college, back in the day. And it cost money to do that. And being poor, he had a really hard time funding all of this education. But he was very good at math, very good at physics, obviously liked biology. But he couldn't afford his education. So what he decided to do is become a monk. Because monks are educated in whatever discipline they want to be educated in. So they have to do the religious side of it, but then they can also pick a subject that they want to teach, because they often taught, and he had to teach early in his career. And so the reason he did this is because they pay for your education. It was one of the major reasons he became a monk. Later he became an abbot, ran a monastery, and obviously before he was an abbot, before he got real busy, he spent all his free time raising pea plants and some other varieties of plants that we won't talk about. So again, I've mentioned most of these, but why did Mendel choose garden peas? Well, first and foremost, because they were there. They're already there, right? I mean, it's nice when you already have them. You don't have to go out and find your experimental subject, right? There were peas. There was a nice pea garden. It was big, large pea garden. And they had already been breeding different variations of the plants there for a number of years. So because of that, and because of meticulous records that monks keep, monks are just very meticulous about what they do. He knew which varieties and which seeds were true breeding and which weren't, based on the number of years they were bred and how they were bred. And so he started with these variants, and he again picked the plants that had these characters that were this or that, this or that, this or that, right? You either had one or the other versions of the phenotype. The other thing about pea plants is they're relatively small, they're easy to grow, they grow really fast, and they have a fast life cycle. So think about if you picked an elephant and it took 50 years before you got babies, something like that. You would never be able to get results. He could get results really quickly, grow bunches of them at the same time, and get a huge data set that he could use to understand how these traits are passed genetically from parent to offspring. The other advantage of using plants, particularly peas, are the sex organs. Both of them are found in each. So you can self-pollinate for your pure breeds and then control cross-pollination, right? By removing the male part of the plant, and you collect pollen, and then you just need a paintbrush, a small paintbrush to do this. And you can collect pollen just like a bee does, and then go over and put it on another plant and grow your own plants. And we've done this, I've done this, you'll do it in genetics maybe, or maybe somewhere else. We used to use fast plants and do the same thing. It's kind of fun to do, actually. Used to do Mendel's experiments and collecting pollen and stuff. It's pretty fun to do. The animal model that most people use when you're doing animals and you wanted to study genetics are fruit flies. So fruit flies, small, easy to breed, get lots of them, get huge sample sizes, and you can do similar types of studies. The only thing is they fly away if you're not careful. There. Pea plants can't run. That's also a major advantage. So here are the 7 characters, at least that he started with. He branched out from this, but these are the 7 major ones that he used. And you can notice with each one, there is this or that, right? There are 2 traits, purple, white, axial terminal, yellow, green, tall, short. Pretty easy to follow the trait. And he experimented on all of these with experimental breeding and found this same type of monohybrid results that we've already talked about with flower color. You can do it with any of these. So one of the first things he used was flower color. We've already kind of went through this because you have purple and white pea plants. So he took seeds from individuals he knew were pure breeding because they've been bred for generations. That is, they've been self-fertilized for generations. The same with white. And he bred those together, right? So they're pure breeding dominant, pure breeding or homozygous recessive and dominant. Took the gametes, pollen, so he cross-fertilized these to get the F1 generation, which were all purple and all heterozygotes, right? And then, the classical part of his experiment is he didn't stop there. He then made those interbreed. So he would take the individuals, again, interbreed them, and get the F2 generation. The only 2 possibilities here, if you have 2 units, is the dominant, get a half of, 50% chance of getting the dominant trait, or a 50% chance of getting the recessive trait. When you do that, you get a 3-to-1 ratio. So for all of those characters, and if you go and look into any genetics book, they'll give you these huge numbers. He bred somewhere between 6,000 and 10,000, or he collected 6,000 to 10,000 offspring for the F1s and F2s for each of those 7 characteristics. So we're talking huge numbers of, way over 30,000 plants to get his ratios. And the ratios always were really close. A matter of fact, most statisticians, we know Mendel was very, meticulous, but his numbers are almost too good. It's like 2.96 to 0.8 to 0.9. They're incredible for all of those characteristics as far as phenotypic and genotypic ratios. So they all look the same. It doesn't matter whether it's purple or white or smooth and round or tall and short. They all the numbers all work out the same. And so that's where his law of segregation came from, and how he knew we now know diploid organisms have 2 copies, right, of the genome. Now, Mendel didn't stop there. He said he thought about this and wanted to test, well, if these segregate independently, each individual gene, we know that organisms have more than one gene. So all genes, or most genes, you didn't say all, but you never say all in science, right? He said most genes are many genes within a genome or within the organism. These units, that control different characters and different traits all must independently assort. And so the law of independent assortment, which you practice with your Martians a little bit, means that if you're looking at more than one gene at a time, they're going to assort independent of one another. Particularly now we know if they're found on different chromosomes, right? Which is what we're assuming, that if you get these numbers and the two genes for two different characters are separating independent from one another, then they're found on different chromosomes. Now, you could follow 2, 10, 20, 50, 60. That would be really hard, right? So we're just going to follow 2. 2 of those 7 characters. Instead of following 1, we're going to follow 2. And so we're going to call it a dihybrid cross. You guys all done dihybrid crosses? Where you follow 2? You could do trihybrids. In your Martians, you were following 5, weren't you? Remember, I didn't make you do all the gamete possibilities because there were like 32 different combinations. We're only going to follow 2 of these at a time. It could be any 2 of those characters, or any 2 characters on a test are going to be a dihybrid cross. And he did the same exact thing. He already knew which of these characters were dominant and which were recessive because he had followed them in monohybrid crosses. Then he went and picked plants that had, dominance for 2 of them, recessive for 2 of them, and followed those together. And did the same thing, bred them, produced huge numbers. So here's a really good example of this. So he picked green pods with yellow seeds, which are true breeding because they've been bred over and over and over again. And he found out this is the dominant for the fruiting body. It's going to be, the wild type is going to be green pod with yellow seeds in this particular species. And the completely recessive will be opposite of that. It will be kind of pale or yellow pods to green seeds. So he took 2 true breeding individuals and bred them, and we know all the offspring are going to be heterozygotes, right? For both traits, and they're both going to be dominant, aren't they? For both traits. And so all of the offspring, the F1 offspring for this breeding, were all green with yellow seeds. And this is what their genotype looks like. Big G, big Y, little y. And you understand why if you look at meiosis, right? Which we're going to do here in a second. So we're going to do a dihybrid cross. And we're going to take these guys now. And I just used AB, all right? We're going to take these heterozygotes, our F2, and we're going to breed 2 of those together. We're going to breed them, right? And when you do that, there are 4 possible gamete combinations when you have 2 heterozygotes. If you had 3 heterozygotes, it's just 2 to the n. 2 times 2 equals 4. If you had 3, it'd be 2 times 2 times 2, which would be 8. If you had 4, so you understand now why I did it in 5, because there's like 32 possibilities. So here we're just going to follow 2. And these are the 4 possibilities. Now you might want to know how, or if you don't remember how to get these, because it can be confusing. It's called foiling. It's really easy to do. And that's on the next slide. This is how you foil. Start with your first trait here, and you simply are going to combine big A with big B and big A with little b. Right? There's 2 of them, right? Then you're going to take the second letter, your little a, and combine it with the little b, AB, and the big B, right? And that gives you all 4 possible gamete combinations. You're going to get that for each of those, right? So that's why you got to do 16 boxes, because you have 4 possibilities for one gamete and 4 possibilities for the other gamete. And then you do a big, huge dihybrid cross. This is the meiosis of it. So instead of 1 chromosome, we're interested now in 2, because these characters, we're assuming, are on different chromosomes. So we just picked, whoever made this, which is a great figure, picked a long chromosome and a short chromosome. The long chromosome has the A locus, the short chromosome has the B locus. You could either have big A or little a. And remember, in this experiment, we're using true breeding individuals, right? And then we got our heterozygotes. So with our heterozygotes, and we're only thinking about 2 homologous pairs, there's only 2 possibilities of alignments. We could put mom on one side and dad on one side, or we could alternate them, right? Mom, dad, dad, mom. And then when we start separating these, you get your 4 different possibilities, like we just foiled with. Big A, big B, little a, little b, and then big A or little b, and little a or big B. Those are the possibilities you can get. All you have to do is work them through meiosis, right? Or foil them. It's easier to do it that way. All right? Yep.
On the slide for that.
One more? Woo.
Can you do like at least one row of examples?
Oh, I do the whole thing here in a second. Yeah. That's the next thing is actually do the dihybrid cross. So I'm going to show you that, yeah. But I wanted to show you meiosis first before we did it. I don't want you to forget about meiosis once you do it, right? All right, so let's do this. So dihybrid cross, we take 2 true breeding individuals, one dominant, one recessive. We get an F1 of all dominant heterozygotes, 100%, right? Or 1, that's what we called it when we did genotype and phenotype, right? Equals 1. Now, if we breed 2 of these, when you look at how many phenotypes there are 4 possible phenotypes. And if these are independently assorting loci, they sort independent of one another, you always get really close to what's called a 9:3 to 3 to 1 phenotypic ratio. If it's not this, like it's 9 to 7, or it's some other number of 16, right? Then they're not independently assorting. You'll learn of genes like sex-linked genes that are linked. They actually travel together. Why? Because they're really close on a chromosome. They're not actually on separate chromosomes, so they travel together. And you'll see that later when you get to genetics. We're not doing that here, all right? You can save that linkage for later on. I hated that when I was in genetics. So this is what it looks like when you do it. So this particular example is flower color and whether you're tall or short. So out of the 16 possible genetic combinations, when you do this, and if you do a huge population of offspring and you count them up, you're going to get a 9:3 to 1 phenotypic ratio. So look at the purple here. In the purple boxes are the 9 dominant characteristics for both traits. So these are purple and tall plants. Tall is dominant, short is recessive. So you get 9 dominant at both traits. Now they don't all have to be homozygous or heterozygous, right? They at least have to have 1 copy of the dominant trait. Then you get 3 in the dark green and 3 in the light yellowish green, I guess. Am I saying that color right? And then 1 pure homozygous recessive for both characters. Now the 3 to 3 is dominant for one trait, recessive for the other. So notice the first one is dominant for purple color, and then recessive. It's short. It's purple and short. This one here is white and tall, right? You see that? It's white for or recessive for the first character and dominant for the second. Then you get just one that's completely recessive, so it would be white and short. Now the genotypic ratio is harder. It's 1 to 2 to 1 to 2 to 1 to 2 to 1. But all you would have to do is count them, right? So there's 1 homozygous dominant for both. There's 1 homozygous recessive for both. And then there's bunches of combinations, right? And you would just have to count them. You could just X them out, if you really wanted to do it. But that is the hardest Punnett square that you will see on the exam. Or unless you want me to do trihybrid cross. Yeah. This is F2. Because it's 2 heterozygotes, right? Now, I could give you an example where you're heterozygous for one and homozygous for the other, and it'll look different than this, right? because it's not two heterozygotes. You just, on the test, just draw the damn Punnett square, right? It doesn't matter. Draw it and count. I hate people that don't, they decide not to draw it and they get it wrong, and I'm like, why didn't you just draw the damn Punnett square and write it out? The other thing that you want to make sure, keep all your A's together and all your B's together. I've seen people put AB, AB, you know, and I'll have a big A, then a little B, and a little A, and then a big B. Put the B's and A's together so you can see whether you have dominant or recessive traits. Does that make sense? As you do it. Also do it in pencil. That's probably a better, you know, all suggestions for the exam. Okay. Yesterday, we were talking about simple dominance and recessiveness, right? We did monohybrid and dihybrid crosses. And then I wanted to give you some exceptions to the rule, just a few. There are lots of exceptions to the rule, but I give you some of the major or more classical examples of differences between this or that, right? That is color versus white, or tall versus short. So these are called codominance and incomplete dominance. And codominance is when two alleles actually, when they come together in the offspring, they are expressed equally or shown equally in the phenotype. Blood typing is a classical or best example of this, where you get antigens or these proteins that are expressed on the surface of red blood cells are given letters. So there's A-type blood, which has the A protein on the surface of the red blood cell, and B-type blood that's on the surface of blood cells, or no protein at all, which we call O blood. And when you get an A and B allele, you express both proteins, right? So you see both of the allele proteins, if you will, on the surface in the phenotype. And I'll show you a picture of that in a second. With incomplete dominance, one allele is partially dominant over the other, or the two interact in some way to give you something really different, a different color, a completely different color. So if you see something in the offspring that is different than the phenotypes of the adults, either one, then you know you probably have something weird like this going on. The best example of this is what they call mixing, when you have an adult flower here that's red, that's cross-pollinated with a homozygous recessive white flower. You would expect all the heterozygotes in the F1 to be red, right? Because you would assume that that red color, like the purple flower, is the dominant allele. But in all the offspring, all the heterozygotes are actually pink. Or sometimes you'll get flower colors that are like yellow, that are completely different than the two adult phenotypes. And then you know the heterozygotes, those two alleles are interacting in some way differently and giving you a phenotype that's much different. That's incomplete dominance. With blood typing, which is also what's called a multiple allele system, where you have more than three alleles that are affecting this. I should say you have genes or a gene with three alleles or more. So you don't just have purple and white, you have here A, B, or nothing, okay? So you can either express the A protein on the surface of the red blood cells, or you can express the B protein on the blood cells. Or you can express neither of those, and that's called O-type blood. The O-type blood is one of the more common blood types, so O negative and positive. So if an A parent and a B parent produce offspring, you'll get AB offspring. So you got codominance. One's not dominating or masking the other, both are expressed equally. So notice with the A-type blood and the B-type blood, you can either be homozygous for big A or homozygous for big B, or you can carry the O allele. See that? Which is no protein. The O allele is recessive, which means if you get an A and an O, you're A. If you get a B and an O, you're B. But if you are kind of almost this quasi-heterozygote, if these two were to breed, an AO and a BO, you might have an offspring that's either AB or O, right? If they get the O allele from both parents. So in this instance, you have not only codominants, but you also have A, B, and O as your kind of your multiple variations in the instructions. Does that make sense? For this type of system. So this is called ABO blood typing. Now, as you're going to come to find, many genes are not controlled by one or even two genes. They're controlled by many, many, many genes with many different allele types. And when you have two or more genes and multiple alleles at those genes controlling a phenotype that you see, particularly if it's a single phenotype like eye color, for example, or height, then you're going to get this continuous variation that are kind of small increments from one another. Eye color is a wonderful example of this. For eye color, there are anywhere between 14 and 150 different genes that can affect eye color. Typically, it's 2 or 3 main genes that really are interacting with one another. But if you have a recessive allele in any of those 3 major ones, then you can get some really interesting variation in eye color. And so these are just a couple of different, eye colors that you can see across the board. Get dominant eye color in the middle, average eye color in the middle, right? And then you'll have variations away from that taper down to 2 extremes, like really light gray blue eyes or really dark eyes on one side. Or height is another good example of this, where you got these small increment or range changes. There's an average height, right, across the male and female human. It's 5'8" in males or 5'9" in males or something is the average height. And then you obviously are going to have really tall individuals like over 7 foot, and then really short individuals under 4 foot, right? And then all the range of variation in between. And notice that they increase as you get closer and closer to the average. Okay, so this is called a bell curve, and that's how most continuous variation pans out. As far as the phenotypes you see are combinations of the genes and alleles that give you an average in the middle and give you this bell curve effect, where most of the population is sitting in the middle of the graph as far as the number of individuals that show that phenotype. So that's what you see with continuous genes. And so a lot of genes, or I should say a lot of phenotypes, are controlled by this continuous type of variation that you see with multiple genes affecting each other. Now, pleiotropy, the last thing I really want to cover, and then I'm going to talk a little bit about the information in our little case study. So pleiotropy is when one gene influences many traits at once. All right, 2 or more, I just say many traits. And I call these genes have a systemic effect on the body. That is, they are being expressed in multiple places at once by different cell types, and it affects the overall function of the entire organism. And some of the more classical or best examples of this are pathologies or genetic disorders. These 2 are really classic. Sickle cell anemia and cystic fibrosis are 2 examples where you get this modification in a gene that affects the entire body, not just one organ or one particular phenotype within the organism. With sickle cell anemia, it's really an incredible disease. You change one base, messenger RNA for hemoglobin, one of the bases are changed, and you get the switch out of a hydrophilic versus a hydrophobic amino acid. And depending on the number of copies of this you get 1 copy of the sickle cell allele and then 1 normal wild type copy, then your cells typically look like this, the red blood cell, like a normal red blood cell. And it's only in a low oxygen or stressful situation where cells sickle and you have a sickle cell episode, which can, for example, at a low oxygen situation. So they might struggle when they climb a mountain above 12,000 feet and all their cells start sickling because it changes the affinity for oxygen and the shape of the hemoglobin protein. If you get 2 copies of the sickle cell disease, then your cells always look like this. And that causes continuous problems as far as circulation, because you can imagine these cells, which have sharp edges, are not only fragile, but they also stick together really easily, and they cause blood clotting. and really lower the life expectancy of somebody who carries both alleles with sickle cell disease. Now, this is very prevalent in certain populations, particularly populations that arose along what's called the malaria belt and along the equator, because malaria is still one of the major diseases that kills lots and lots of people yearly. And if you carry one copy of the sickle cell and one normal copy, so you have normal cells typically, you are resistant to the malaria bug. So it's has some really good evolutionary consequences or advantages in these malaria belt areas. The problem is if you have parents that carry this recessive allele and they give it to their kids, then they're going to have full blown sickle cell all the time. So it affects many body systems or many organs across your entire organism. Cystic fibrosis is also similar to this. So it has this pleiotropic or systemic effect. Cystic fibrosis is when you have a gene modification in mucus-producing cells, the cells that produce mucus. And so where you can imagine, think of all the places in your body where you produce mucus, right? The digestive tract, the reproductive tract, the respiratory tract. All of those cells which are using this gene are going to produce a really thick mucus instead of normal mucus. And that has a tremendous effect on the entire body. Okay, so that's what I mean by pleiotropy. So it's kind of this change in one gene that really affects the entire body. There are a lot of other examples, but those are two of the more classical examples. All right, questions on any of that? That's really where we had to finish up. The one other thing I really want to talk about before we get into our case study, what I'll do is turn that off and is sex-linked traits. So we're going to talk about these Hawaiian crickets. And so this is a really wonderful example of evolution happening in real time on islands in Hawaii. One other really interesting example of genetics that differs from dominance and recessive are sex-linked traits. And so sex-linked traits are most commonly for us are going to be carried around with the X chromosome. Okay, so in us, for example, XX is the female, right? And XY is the male. And because we males, I should say the males in the room, only have one X, if we carry a recessive trait for a mutation that's either an advantage or a disadvantage, we will express it. So if you have a recessive trait, you express it because there's not another X to mask it, right? Whereas in females, they have two X's, and so they can mask or not show the recessive phenotype. So these X-linked traits are often common in males and very rare in females. And so we're going to have this example with insects because insects don't have a Y. They simply don't have another X. They just use a 0. And the females are XX. So when a male inherits an X, or he is male, he only gets one copy of the X. And so it's kind of like the XY system. If you have the recessive traits, then you're going to show it. Where the females will typically either be heterozygous or homozygous dominant and never show it. Okay? So we're going to be looking at something that crickets do. Male crickets, they sing, right? They sing by, do you guys know how crickets sing? You know, at night when you hear the crickets singing, they use their wings, right? They rub their wings together. And so their wings have structures on them that are called the harp and the window. And the harp and the window, or they call it the mirror, harp and the mirror. Those two structures are kind of make the wing. We think of a typical cricket wing as my hand will be really thick. And so when they rub the wings together, it amplifies the vibration. So you hear the cricket. They're going to have a phenotype here that's called a thin cricket, or what they call a flat wing cricket. And their wing is really flimsy. And so when they try to, produce sound using the file. The wing's not going to vibrate very much, so it's not going to resonate any sound. Not only that, their file is pretty much absent. And so they'll show this in the first figure there that you see in your text. And what they're going to test is what type of genetic control of this gene in the males that you see, because the males are the ones that sing. And since you don't see singing in females, one might hypothesize that it's on the X chromosome, right? Or if it's not on the X chromosome, then it's an autosomal mutation, which are all other chromosomes if you're not a sex chromosome. And then you have simple dominance and recessiveness, right? It's a dominant trait, and as long as you carry one big letter, right, you're going to express it. For a recessive trait, you'd have to have both recessive traits, right, in order to see it. We don't care whether it's male or female because all the males are going to have either, be dominant, or all of the males are either going to be recessive when you're talking about whether they express the trait or not. So it'll follow the typical dominance and recessiveness we talked about. So you're going to have to do Punnett squares for the 3 of these. When you look at the Punnett squares of these, you're simply going to use the normal ratios like 3:1 or 1:3 or 1:2:1, right? When you talk about sex length, we only care about the males, right? Because the males are the ones that are going to express singing or not. And so we only count the males in our Punnett square. You ignore the females, okay? Does that make sense? Because we don't care about females when we're talking about sex. And sex is controlled by the X chromosome. And hopefully that'll help you a little bit with sex linkage. So does this kind of make sense? Kind of how things travel through the X chromosome? It'll give you different ratios. So instead of 3:1 or 1:3, you'll see 1:1 or 50% with the males. You'll see that when you start doing your little Punnett squares. The other thing that I think will help it, and what this leads really nicely into, is that we're going to start talking about natural selection, or what we call, the main driving force of evolution. And if you remember, I talked about how genetic variation is really important to drive evolution, right? Natural selection is kind of how evolution happens. It's when nature uses something called a selection pressure. That's the way I like to teach it, at least. So a selection pressure to choose which phenotypes are going to survive and which are going to die out, or which have an advantage or an adaptation versus ones that don't. These selection pressures are things like predation, right? Or, in our case, parasitism. So there's going to be a new fly that comes into the Hawaiian Islands that parasites crickets. And it's going to dramatically affect what happens. And so when you look at, I think it's figure 2, it's a graph that shows years over number of singing males. And when the scientists started studying this group on the islands, there were all these singing males, right? And then they came back 2 years later and there were no singing males. None at all. Where do they all go? Then they came back 2 years later and then there are a bunch of males again, but none of them sing. So that had them start studying, well, what's going on here? Why were there no males? All the males were gone, and then also we have males that don't sing or are called silent males. And then they show you pictures of these, and then add this parasitic fly that was a new selection pressure. So when a new selection pressure comes into an environment, it is going to put more pressure, right, on certain phenotypes. This is why having many phenotypes within a population is advantageous, right, for evolution, because you never know when a new selection pressure is going to come in. So they were singing, and the singing males were doing fine because females like males that sing, until a parasite comes in and kills all the singing males. And then only those that don't sing are the ones that are actually breeding. And then a new phenotype comes in called purring a few years later that helps with males breed with females. So remember, phenotypes are controlled by genotypes, right? So the more genotypes we have, the more types of alleles that we have, or the more variation that you have in a population, the more phenotypes you have, the better the population will deal with these selection pressures. This is why genetic variation is really important. The other major part of this is what's called sexual selection. Right? This is a different type of evolution where there is mate choice. Your mate chooses what he or she wants to breed with. The most common types of mate choices for us is female choice, where the females are choosing males based on some phenotype. Okay? The phenotype here is singing. So crickets that sing loud and proud have good genes. So the females are using a phenotype to determine genotypic strength. So males that sing the way it goes have to avoid predators, have good immune systems, use a lot of energy for the singing process, or have good metabolism, right? They're able to do all this and survive and still reproduce. So females love, right, in this instance, crickets that sing really well. Obviously, ones that are silent, they don't like to breed with. So you're playing basically in this scenario, tug of war between these two, right? There are obviously advantages to both and costs to both. And it's a tug of war between the two. You bring in the new parasite, singing really doesn't matter that much, right? Because there are going to be no singing males. And then the big question is, well, how do silent males, breed? How do they compete? And they explain kind of how that happens in what's called satellite breeding. And then what is the purring cricket? The purring cricket that starts showing up on islands is going to be basically the compromise, right? Whenever you're playing tug of war, if you want both sides to win, then you got to meet each other in the middle, right? And that's what purring does. Purring is going to be not full-blown singing, so loud enough for females to hear, but quiet enough to escape parasitism. All right, so it's an awesome example of how evolution works real time.